So now you can advance because this is me happy in the lab, and then the two next slides is just the titles that you've said before. So I'm going to present you the part 2, which is on immunology. And can you press two times, Eleni?
Yes.
On immune response, digestive enzymes, and antioxidant status. I'm responsible for the immune response studies, and my colleague Efi Kotu did all the analysis of digestive enzyme and antioxidant status. So Eleni can, yeah, okay. So those are the 5 diets that Eleni described to you. I've just put in different colors so that it's easier to understand the results. So the positive control is in blue with the fish meal, the fish that were fed with the fish meal-based diets. The plant meal-based diet is in green. Then you'll see the heat-treated diet in orange. The plant meal diet supplemented with solid-state fermentation products in purple, and in yellow, the plant meal diet where phytase and xylanase were added to the plant meal, the 3 different plant meals. We took 3 samples of serum for the analysis of the immune and inflammatory parameters, 5 liver samples for the oxidative enzyme analysis, and 5 gut samples for the digestive enzymes. Can you press, Eleni?
Yes. It don't goes on. Oh my God, what happened? Κόλλησε.
Oh.
It don't goes on. Okay. This, so, I hope this is the 3rd one, eh?
Yeah, it's good, it's good.
Okay.
So this is the antibacterial activity of the serum. So on the left here, you can see the lysozyme activity, which is the antibacterial activity of the serum against gram-positive bacterium. And you can see that the control negative fish are all showing lower lysozyme activity than all the different diets, and especially from the fish meal diet, which is not significant. But you can see that the addition of enzymes, either solid-state fermentation products or phytase, xylanase enzymes, those both increased to the level of fish meal fish, or even further those fish for the lysozyme activity, and the heat-treated also reached even higher levels than the fish meal fed diets, fish. When you go to the 2 graphs on the right side, those are both looking at the complement activity. The one in the center is looking at the 3 pathways of complement, and the one on the right is looking at the alternative pathway of the complement. So when you look at the one in the center, you've got, again, immunosuppression of the complement activity in fish-fed plant meal diets. And this suppression was reversed when you added the solid-state fermentation products.
Yeah.
It was also a little bit better in the heat-treated control diets, and no difference in the phytase xylanase added diet. When you look at the alternative complement pathway, you can see that it doesn't, it's not so different from the first graph, because most of the complement activity is done by the alternative complement pathway. But you can see that the difference between the 2 graphs is the classical pathway and the lectin pathway. So the classical pathway being based on the complex between antigen and antibodies. So you can see that the main difference between those 2 graphs are in the solid state fermentation, when you can see that it's lower when you're looking only at the alternative pathway. So we can suggest that the solid state fermentation products do stimulate the classical pathway of the complement. So you can press several times because there were some red circles showing where but yes. Anyway, it's not so easy to do at distance. So let's go to the next slide. This is again some immune parameters. So we were looking here at the reactive oxygen species production and the inflammation. As you may know, the activity is transforming hydrogen peroxide into a very toxic form of hydrogen oxide with linked to the chloride ion. And when it stays inside the neutrophil and there is no too much expression of this myeloperoxidase, it's got a very nice antibacterial activity, and it's going to kill your pathogens. When it gets out of control, it gets out of the immune cells, and then it can become oxidative to the fish cells. And then you've got inflammation and you've got problems going with it. So you need an equilibrated reaction there. So when you see there was no significant difference either in the ROS production or in the inflammation on the right. But you can see that there is a tendency again for suppression of in the negative control diet and reversion to the levels of fish meal fed fish, with the addition especially of solid state fermentation products. And this was the positive side of this effect was underlined by the graph on the right, which shows that there was no problem with inflammation when we measured the positive acute phase protein, which is ceruloplasmin. We can go to the next slide. So let's go to the antioxidant defense system enzymes that were measured in the liver. So again, going from hydrogen peroxide, which is produced by the fish by the xanthine oxidase enzyme, from molecular oxygen to reactive oxygen species, when the fish are exposed to stressful environment, or like UV, or when there is a cytokine message, or when there are growth hormones or disease or drugs or pollutants, whatever, many ROS, reactive oxygen species, are produced, and the fish needs to detoxify them so that there won't be any negative effect on the fish cells. So those enzymes in the liver, like catalase, glutathione peroxidase, and glutathione S transferase, they transform this hydrogen peroxide into molecular water. So there won't be any toxicity anymore. So when you look at all those three enzymes that were measured in the liver, you can see a very drastic effect of the plant meal on the catalase activity when we compare it to fish meal fed fish, which was a little bit reversed when we used the enzyme on this plant meal or when we heat treated this plant meal. It was significantly better, but we didn't reach the fish meal levels. When selenium-dependent glutathione peroxidase, we had the same image, the same picture from this, which means reduction in negative control diet with reversion of, increase again in when they were added with solid state products or heat treated, but no effect of phytase and xylanase. And again, looking at glutathione S transferase, solid state fermentation product were the best to reverse to fish meal level when our diets were poor in fish meal and rich in plant meal. You can go to the next slide. Yeah, again, all the never mind. So those are the digestive system enzyme in the gut. So we looked at trypsin and aminopeptidase activity, and we've got the same image with reduction in the negative control, increase when solid state fermentation products were added, a slight increase for the heat treated plant meal. Concerning trypsin activity, it wasn't evident anymore concerning the aminopeptidase. And phytase and xylanase were even reduced the trypsin activity, which I could only think of one potential explanation, which could be that the phytase xylanase do disrupt the membrane of some bacteria that are in the gut that would help the fish with the trypsin activity. That's the only explanation I could think of, but there may be another one. So we can go to the next slide, which is a summary of all those effects on the health. So you can see on in the first column the effect of the negative control compared to the positive control based on fish meal. So mainly almost all of the parameters that we looked at least the ones that I presented, were affected by the diets, by the plant meal based diet. And in the three next columns, you can see when there is a dark color, you've got significant difference with the negative control. And when there is a light color, you've got a tendency for improvement of this particular parameter compared to the negative control. So you can see that basically solid state fermentation improved almost all of those parameters, including immune parameters and antioxidant enzyme and digestive enzymes. Heat treatment was not so efficient on the immune parameters. There was a tendency, but no significant difference. But there still was a nice, very strong activity on the antioxidant enzyme and one digestive enzyme. And phytase and xylanase were not so efficient, and they showed significant differences only in two of the parameters tested, one immune parameter and one antioxidant enzyme. So basically from the health, you can see a different picture than what was seen in the growth. When we look at the bibliography, it hasn't been so much studied on the health benefits, but we've seen that solid state fermentation was also shown in carp and in tilapia to improve gut and liver histology. Heat treatment was shown to restore lysozyme activity when fish were fed soybean meal protein compared to soybean meal low in trypsin inhibitor. There was no fish meal control in this study. And phytase was, when on its own, significantly improved the immune response of Nile tilapia. And xylanase, I found one study which showed that together with glucanase, it significantly increased digestive enzyme activity in the posterior intestine of turbot. So in conclusion, if we go to the next slide, solid state fermentation product provided the fish with a cocktail of enzyme produced by a fungus that may act in a synergic manner and thus may improve further the action of the mixture of exogenous enzymes of different origins. This needs to be further studied, of course. We don't know it, but this is a supposition. In the present study, solid state fermentation products showed the best potential for immunostimulation and improvement of antioxidant status and digestive enzyme compared to the cocktail of phytase and xylanase. Those enzymes degrade non-starch polysaccharides, thus minimizing their immunosuppressing activity, and degrade phytate, thus freeing phytate-bound minerals and nutrients that may improve the antioxidant status. And the heat treatment of plant meal also improved the immune and antioxidant status of the fish and some digestive enzymes, probably through the degradation of antinutrients, such as trypsin inhibitors or tannins or other ones. And I thank you very much for your attention.